A very good morning to all of you. So in today's video, I'll just sharing some important question for the Viva, Grand Viva in the university exam. Okay, and this video is mainly for the UG students. For PG, that is the MD Biochemistry exam, I'll share some another video. Okay, so for UG students, so let's start the topic. And what the things you must read before your Viva, Grand Viva exam in your practical university practical. Okay. So first thing that you have to study is in cell. In cell, the important thing is you should know the various marker enzymes of all the organelles. First thing, okay. What is the marker enzymes of all kinds of organelles? Second important, Zellweger syndrome. What is Zellweger syndrome? And next on the plasma membrane structure, fluid mosaic model. What is fluid mosaic model and why fluid mosaic model is known as fluid mosaic? What is the reason behind that? Why the scientists name that model as a fluid mosaic? Third important, the name of scientists who proposed this fluid mosaic model. Okay. Now comes on the transport. In transport, important is that of you should know the difference between active transport and passive transport. Okay. What is facilitated diffusion? What is difference between your primary active transport and your secondary active transport? Next important is that of what is receptor mediated endocytosis and give the example where this receptor mediated endocytosis will occur inside the body. Okay. Next important thing is that what is the role of glucose in ORS solution? It's very important for your Viva point of view. Okay. Next is that of your ion channels. What is difference between the voltage gated ion channel and your ligand gated ion channel? Ion channels should know, and another thing is you should know the at least 2 examples of the ion channel disorders, that is of ion channelopathy. Okay, so in ion channels you should know the difference between voltage gated and ligand gated ion channels, and the example at least 2 disease you must know for the ion channelopathy, that is the disorder related with your ion channels. Okay, all right. Next comes on the enzymes. Okay, enzymes you should know classification. Okay, what is the classification of enzymes? Okay, and what is the significance of that EC number? While you will write the classification of enzyme, you will write 4 number of EC number. Okay, and while you will write like SGOT. Suppose so the EC number of SGOT is 2611. So what these 4 numbers will signify? What is the significance of these 4 EC numbers that you have to know, and you have to know the classification of enzyme and each class along with their examples. Okay, at least 2 examples of each class you have to know. Okay, important difference between lyases and ligases. What is difference between lyase and what is difference between ligase? Okay. And what is difference between synthase and synthetase? Alright, difference between lyase and ligase and difference between synthase and synthetase. Alright, next is that of important is that of lock and key model. Who proposed lock and key model and what is this model states? Lock and key model. Second, Koshland theory. Who proposed this Koshland theory and what is inside this statement of Koshland theory? Okay. Next, important difference between coenzyme, cofactor and prosthetic group. Okay, the difference you must know: coenzyme, cofactor and your prosthetic group. Alright. Now, another important thing is that factors affecting enzyme activity. At least you should know the name of all factors that affecting the enzyme activity. Next important, what is Michaelis-Menten constant, Km. Okay, what is Km? How you will define Km? How you will, or your examiner can ask how you will define the Michaelis-Menten constant Km, and what is the significance of Km in the enzymes that you have to do. Next, enzyme inhibition. You know the example of each and every inhibition, like competitive inhibition, your non-competitive inhibition, your uncompetitive inhibition, and societal inhibition. Okay, all of these inhibition you must know what is the changes in the Km and Vmax, and the example of at least 2 to 3 example you must know all inhibitions. Okay, in suicidal inhibition must read what is the mechanism of action of allopurinol. Okay, and why the allopyridinol inhibition is called suicidal. What is the reason behind that? That you have to know. Okay. On KM and VMAX, already I shared mnemonic, so you can watch that, and the Lineweaver bug plot. Here I also share the trick on my channel. You can go through that. So how you will recognize if there is a cross, then what Lineweaver bug plot say, and if you are V or if they are parallel lines, then what is the Lineweaver bug plot say. And Lineweaver bug plot is very important for your spotting also. So that is must read Lineweaver bug. Okay. And next important is isoenzyme. What is definition of isoenzyme? You must know exact definition of isoenzyme and what are the examples of isoenzyme. Okay, and the ALP, different isoenzymes of ALP, different isoenzymes of LDH, and different isoenzymes of your CK, creatine kinase. Isoenzymes is a must-read topic. Another important thing in isoenzyme is that of your flipped pattern for LDH. Okay, what is flipped pattern which occur in LDH? So you have to know idea about flipped pattern. Okay. Next important is that of diagnostic uses of. Okay, what is the diagnostic use of enzyme? What is therapeutic uses of enzyme? And what is the laboratory uses? So diagnostic, therapeutic, and laboratory uses of enzyme is must read. Okay, you have to read the like your this thing. Okay. Now comes on the your carbohydrate chemistry. So, and one important thing in that is that of enzyme, that mechanism of enzyme action. You just simply know what is the role, how enzyme, what is the role of enzyme in activation energy. Okay, how enzymes will play role, and how the enzyme act as a biocatalyst. So activation, what is the impact of enzyme on your this thing, activation energy, and why the enzymes act as a biocatalyst. And the other is like all enzymes are proteins except which is the enzyme which is enzyme, all the enzyme, but is not proteinaceous in nature. So that is your riboenzyme. Okay, riboenzymes are not proteinaceous in nature, although they are enzymes. That you have to. Okay. Now comes on the carbohydrate chemistry. Okay, carbohydrate definition. A proper definition of carbohydrate you must know. Okay, there is no guts and in this definition. Okay, as such definition written in your book, you have to mug up that. Okay, so definition of your carbohydrate, classification of carbohydrate, definition, classification, and in classification you must know the examples of monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Okay, you have to know. Homopolysaccharide, now comes on the polysaccharide. To homopolysaccharide and heteropolysaccharide, you must know the examples of homopolysaccharide, you must examples of heteropolysaccharide. Now comes on your heteropolysaccharide. Heteropolysaccharide example, and in heteropolysaccharide most important is that GAGs. What is glycosa aminoglycans or mucopolysaccharides? I already shared the video, watch that. Okay, and the question is asked, why these mucopolysaccharides are known as mucopolysaccharide? Okay, why the name mucopolysaccharide given to this? Next question is asked, why these GAGs are slippery in nature? What is the reason behind that? Why they are slippery in action or why they are slippery in nature? Okay, and you should know composition of each and every mucopolysaccharide. Next important is that of which GAG doesn't contain the uranic acid. Okay, you should know keratin sulfate is the only GAG which don't have uranic acid. And composition, in which GAG there is a glucosamine, in which GAG there is a galactosamine. For that I also shared the mnemonic. Okay. Next important is glycemic index. What is glycemic index and what is the significance of glycemic index? You have to know. Okay. Now comes another thing is that of in isomer part, you have to know what is definition of isomer, what is functional isomer, okay, what are epimers, what are anomers, and what are the two epimers of glucose. Okay, you have to know what are the two epimers of your glucose. And next thing, important thing you have to know is the mutarotation. What is mutarotation? What do you understand by the term mutarotation? And what is the clinical significance of mutarotation? Okay. Next important is why sucrose is known as invert sugar. Now next another thing is why sucrose is non-reducing in nature. First thing you should know which is a non-reducing sugar like sucrose is non-reducing, and what is the reason behind that? Okay, why sucrose is non-reducing. Okay, so this is all about your chemistry of carbohydrate. Now comes on the metabolism of carbohydrate. So in carbohydrate metabolism, important thing you know, and glycolysis, you should know the energetics. Okay, what is the energetics of glycolysis and the regulation of glycolysis? The important thing in all metabolism, you must know in your grand viva the rate limiting enzyme of each and every cycle of your biochemistry metabolism. Okay, so you must know the rate limiting enzymes of all the cycles of your biochemistry. I also shared that video. Let's watch that. Okay, so you must know in your grand viva rate limiting enzyme of each and every cycle. So come back on the carbohydrate metabolism. Glycolysis, important is energetics and the your regulation. What is the role of phosphofructokinase 1? Importantly, how phosphofructokinase 1 regulated the glycolysis. Important is difference between hexokinase and glucokinase. Okay, what is the difference between hexokinase and glucokinase? Next important is what is Rappaport-Levering cycle and what is the significance of this Rappaport-Levering cycle? Or examiner can ask what is the role of 2,3-BPG in your body? Okay, and how this 2,3-BPG is produced inside the body. Question can also be asked like that. Okay. Now comes on the PDH complex. What are the different enzymes and coenzymes of your PDH? That is your pyruvate dehydrogenase complex. So you must know all the 5 coenzymes of the PDH. In TCA cycle, you must know the energetics and the rate limiting enzyme. Okay, so what are the energetics of your TCA cycle? What are the rate limiting enzyme of your TCA cycle? Important thing, the amphibolic role of TCA cycle and anaplerotic role of TCA cycle. Okay. In TCA cycle, you must know amphibolic and anaplerotic role of your TCA cycle. Now comes on the HMP shunt. HMP shunt, the important thing you should know in your Viva is what is the significance of HMP shunt. Nobody will ask the entire cycle in the Viva. Okay, that is for your theory purpose. So in Viva, you must know the significance. What is the significance of the HMP shunt? Second thing, you should know what is the role of NADPH inside the body. Okay. What is the role, or what is, you can say, what is the significance of NADPH in the, you have to know. For NADPH, you have to know, another thing you must know, respiratory burst. When you will tell you the answer, then in NADPH, important thing you should know is respiratory burst. What is respiratory burst and how NADPH play a role in a respiratory burst. Okay, alright. Next important is G6PD deficiency. What is glucose 6-phosphate dehydrogenase deficiency? In which cycle this G6PD deficiency is related? What are the different risk factors of the G6PD? And what is the role of anti-malarial drug in G6PD? Okay, if patient is taking primaquine, then why he is more prone to develop the G6PD deficiency? Explain. So you should know the reason behind that. Okay, G6PD. Next important question, why alcohol causes hypoglycemia? Why alcohol causes hypoglycemia? In glycogen metabolism, you must know the glycogen storage disorder. All glycogen storage disorder you should know on their serial order with their name, with their enzyme deficiency, and what are the important clinical features associated with the all glycogen storage disorder. I already shared the video with all tricks. If you can watch, you just go through that. Okay, and the important thing in glycogen storage disorder is that of importantly, you must read von Gerke's disease. Okay, why von Gerke's disease occur and why in von Gerke's which enzyme is deficient and what is the reason behind the clinical feature of your von Gerke's. Why there is a severe fasting hypoglycemia in von Gerke's. Why there is doll-like facies in von Gerke's. What is the reason behind hyperuricemia in von Gerke's and what is the reason behind lactic acidosis in von Gerke's disorder. Okay, so you must know the reason during your Viva. What is the biochemical basis or what is the reason behind that? Important thing is that which lysosomal disorder is also glycogen storage disorder. Okay, so which or examiner can say which glycogen storage disorder is also lysosomal storage disorder. So the answer is that of Pompe's. Okay, Pompe's is only glycogen storage disorder, which is also your lysosomal storage disorder. Okay, and important, which organ affecting the Pompe's. Okay, next important is in McArdle's, why there is exercise intolerance in patients of McArdle's disease. Okay, important is why, what is second wind phenomenon, and where in which glycogen storage disorder this second wind phenomenon will occur. That is McArdle's. Okay, so I have already shared all these features. You must watch that video of your one garkis and glycogen, because it's a very important topic for your grand Viva. Okay. Next is that of your gluconeogenesis. So you should know the definition of gluconeogenesis. Okay, how you will define the gluconeogenesis, and you should know at least brief how the new glucose is synthesized from pyruvate, from your lactate, and from your hot chain fatty acid, that is your propionyl CoA, and your alanine. Okay, from these four at least you have to know, from pyruvate, from lactate, from your hot chain fatty acid, that is of your propionyl CoA, and your alanine, and how this what are the regulatory enzymes of this gluconeogenesis. Important thing, in which state gluconeogenesis will take place, whether in fed state or in fast state, and which hormone regulate the pathway of your gluconeogenesis, whether insulin or glucagon, that you must know. Okay, an important thing that you know, another thing is that of your in galactosemia, you must know for Viva classical galactosemia. What is classical galactosemia? What are the features of classical galactosemia, and which enzyme is deficient in classical galactosemia? And second important thing, why there is a cataract? Why the patient of galactosemia patient present with the cataract? What is the reason behind that? Okay, so you should know why the patient of galactosemia present with the cataract and what is the reason. Okay, now your diabetes mellitus. Diabetes mellitus, you should know difference between type 1 and type 2 DM. Okay, and you should know the diagnostic criteria of DM. And in diagnostic criteria in Viva, you should know both diagnostic criteria. WHO criteria, what is WHO criteria for diagnosis of diabetes mellitus, and what is your ADA, American Diabetic Association, what is ADA criteria. Okay, WHO as well as ADA, both criteria for diagnosis of diabetes mellitus you should read. Important thing is that what is the complications of diabetes mellitus. Okay. Important thing, DKA. What is diabetic ketoacidosis and management of DKA? If patient comes in your emergency, you are being a doctor, you are posted in emergency, and the patient of DKA comes, then how you will manage that patient. Okay, so you should know DKA, and you should know the treatment or management of your DKA. Okay, so diabetic ketoacidosis is important. Okay. So this is all about your carbohydrate metabolism. Now comes on the lipid chemistry. Definition of lipid. You must know what is, how you will define lipid, and what is the classification of lipid. What comes under simple, what comes under complex, derived, and your miscellaneous. So you should know the classification of lipid. Another thing, you should know the phospholipids. What is phospholipid and how phospholipids are classified. What is sphingophospholipid? Okay, you know the composition of your sphingophospholipid. You should also know what is lecithin, what is cephalin. Okay, lecithin and cephalin. Other important is that of respiratory distress syndrome, RDS. That is also known as your hyaline membrane disease, that is HMD. So whether RDS or hyaline membrane disease, both are same thing. So you must know what is respiratory distress syndrome, what is the reason behind that respiratory distress syndrome, and how you will treat the patient of respiratory distress syndrome. What are the clinical feature child presents if suffering from respiratory distress syndrome. An important thing is what is significance of LS ratio. Okay, what is the significance of LS ratio in your respiratory distress syndrome? And important thing, what is the finding in chest X-ray? There is a ground glass appearance if you will take the chest X-ray of this respiratory distress syndrome patient. Okay, so what is the chest X-ray finding of your respiratory distress syndrome? Next, important on the fatty acids. Fatty acids, important thing, you know, the classification of fatty acids. How you classify fatty acid. Another important thing is that of essential fatty acids. What are essential fatty acids? And name the 3 essential fatty acids. Another important is that of what is semi-essential. As linoleic, linolenic, arachidonic, both are 3 your essential fatty acid, and out of this, out of 3, which is the semi-essential, and why that is semi-essential, why that is not essential. So you should know the reason behind that why arachidonic acid is that of your semi-essential, and what is reason behind that why it is semi-essential. Important, if there is a deficiency of this essential fatty acid, then patient is suffering from what kind of clinical features. Okay, next is your eicosanoids. Okay, what are eicosanoids and from which precursor these eicosanoids will synthesize inside the body? Important thing, what is the clinical significance of prostaglandin? Okay, and what is the mechanism of action of your aspirin and NSAIDs? Okay, at least at your UG level, you should know the mechanism of action of aspirin as well as mechanism of action of your NSAIDs, non-steroidal anti-inflammatory drugs. Okay, their mechanism of action related to your eicosanoids. Lipoprotein, you should know the classification of lipoprotein. If you will do the electrophoresis of lipoprotein, then what will you get from cathode to anode or anode to cathode? From both sides you should know. Okay, and which is the largest chylomicron, which is the smallest chylomicron, which is high density, in which lipoprotein contain highest amount of protein, and which is known as good cholesterol, which is known as bad cholesterol. Okay, so these things at least you should know. Okay, so that's all for today. In next video I'll share another important topics regarding your grand Viva. Thank you.